The following scenario is hypothetical and presented for educational
It started as
and
a simple
entertainment
scientific
purposes.
experiment.
That is what the official records say. In the late 1960s, when lab coats were whiter, ethics boards were thinner, and optimism about the space age bordered on religious fervor, researchers began asking a peculiar question. What is the minimum requirement for life? Not philosophically, not poetically, biophysically. Strip away oxygen, water, pressure, radiation shielding, what remains? Enter the unlikely protagonist, the tardigrade. Known more affectionately as the water bear, this microscopic invertebrate looks like a gummy bear designed by a committee of biologists with a questionable sense of humor. Stubby legs, vacuum cleaner snout, a gait that suggests it's perpetually late for something. Harmless, cute even. Which makes what happened next deeply unsettling. When scientists exposed tardigrades to extreme dehydration, they didn't simply die, they shut down. Their metabolism dropped to undetectable levels. Water content fell from roughly 85% to near zero. Proteins folded into protective configurations. DNA condensed. Cellular structures vitrified, literally turning into a glass-like state. It was less survival and more biological time travel. This state is called cryptobiosis. Think of it as nature's version of airplane mode, except instead of silencing notifications, you silence entropy. Then came the radiation tests. Ionizing radiation at doses that would shred human DNA into molecular confetti barely slowed them down. Vacuum exposure? Fine. Temperatures near absolute zero? Mild inconvenience. Brief exposure to temperatures above 150 degrees? Not ideal but manageable. Eventually, someone had the audacity to put them into space. In 2007, during the Photon M3 mission, dehydrated tardigrades were exposed directly to the vacuum of space, cosmic radiation and ultraviolet flux included. Many of them survived. Some even reproduced afterward, as though space had been a mildly inconvenient camping trip. Science said, fascinating. But buried in obscure lab reports and conference proceedings was something stranger. A pattern. Subtle, statistically inconvenient, and quietly dismissed. Specimens exposed to prolonged radiation didn't just survive. Some showed elevated expression of unique proteins, like Dsup, short for damage suppressor, that actively shield DNA from breaks. That alone was revolutionary. But here's the detail that kept one unnamed researcher awake at night. In certain trials, post-exposure metabolic rates spiked above baseline, as if something inside them had not merely endured the stress, but adapted to it with enthusiasm. Science says this is a myth, that we are anthropomorphizing resilience, that extremophiles are just well-adapted organisms, not harbingers of anything dramatic. But the data says otherwise. Because if an organism can suspend death, defy radiation, and reassemble itself after space exposure, what exactly defines its limits? And what happens if those limits were never where we thought they were? The experiment was archived. Funding shifted, the world moved on, but one footnote remained, almost invisible. A line about anomalous metabolic persistence. No follow-up study was ever published. And that is where the nightmare begins. Now start part 2, the physics of refusing to die. If you want to understand why the tardigrade is unsettling, you must begin with thermodynamics, specifically the second law. Entropy, the slow, relentless slide into disorder, is not merely a suggestion, it is the universe's favorite hobby. Stars burn out, atoms decay, leftover pizza molds with terrifying efficiency. Life at its core is a temporary rebellion against entropy, sustained only by constant energy input. The tardigrade, however, appears to negotiate with entropy like a seasoned diplomat. When dehydrated, it enters the ton state, a shriveled, barrel-shaped configuration that looks less like a living creature and more like a biological raisin. In this form, metabolic activity drops to nearly zero. Not low, not slow, nearly zero. Oxygen consumption becomes almost undetectable. ATP production flatlines, and yet the cells do not collapse into chaos. The trick lies in vitrification. Instead of allowing ice crystals to form, which would rupture membranes and shred organelles, the tardigrade replaces water with trehalose and specialized intrinsically disordered proteins. These proteins solidify into an amorphous glass-like matrix. Molecular motion halts, chemical reactions pause. Time, biologically speaking, hesitates. This is not survival in the traditional sense. This is strategic suspension. Now consider space. The vacuum of space is not simply empty. It is a violent absence. Pressure drops to near zero, causing water to boil at room temperature. Unshielded ultraviolet radiation slices through molecular bonds. Galactic cosmic rays introduce high-energy particles capable of snapping DNA strands like brittle twigs. Humans in that environment would perish in minutes. Unprotected bacteria would fare poorly. Yet dehydrated tardigrades endure days. Physics tells us radiation damage scales with exposure and vulnerability. Biological systems are especially vulnerable because DNA encodes continuity. Break the strands, scramble the bases, and replication collapses. Except tardigrades possess Dsup, a protein that binds to nucleosomes and reduces DNA damage from X-rays by up to 40% in experimental models. Insert the Dsup gene into human cultured cells, and those cells demonstrate measurable radiation resistance. Pause there. A protein evolved by a millimeter-long invertebrate can enhance the radiation tolerance of human cells. That is not myth. That is published data. Now return to those anomalous metabolic spikes after radiation exposure. In several overlooked datasets, post-cryptobiotic revival showed increased expression not only of Dsup, but of stress response genes associated with repair and cellular restructuring. For a brief window, the organism's molecular machinery appeared hyperactive, as though it were not merely repairing damage, but refining itself. Adaptation under stress is normal. Evolution thrives on it. But here is the uncomfortable question. Evolution typically requires generations. The tardigrade seems to compress resilience into a single lifetime. Imagine an organism that can repeatedly endure catastrophic stress, accumulate minimal damage, and return each time with reinforced protective pathways. Not immortality, something subtler, something iterative, a biological feedback loop against destruction. Mainstream science reassures us. They're extremophiles, nothing more. The mechanisms are biochemical, not mystical. Glass transition temperatures, protein stabilization, efficient DNA repair pathways. And yes, that is all true. But physics also tells us that systems exposed to repeated high-energy stress tend to degrade. The tardigrade does not degrade, it persists. And persistence under the wrong circumstances can become something else entirely. Because if a creature can survive vacuum, radiation, freezing, desiccation, and return metabolically invigorated, what would happen in an environment less hostile than space? Say, for instance, a warming planet increasingly saturated with radiation sources and environmental extremes. That is when the conversation shifts from curiosity to consequence. And that shift is where our first major climax quietly approaches. Because someone decided to test not just survival, but enhancement. Part 3, the enhancement trials. There is a particular moment in every ambitious research program when curiosity crosses a line and becomes temptation. For tardigrade science, that moment arrived when researchers stopped asking, how do they survive? And started asking, can we use this? It began innocently enough. After the discovery that the Dsup protein could reduce radiation-induced DNA damage in cultured human cells, laboratories around the world began replicating the experiment. Insert the tardigrade gene, expose cells to X-rays, measure survival rates. The results were consistent. Human cells expressing Dsup showed significantly less DNA fragmentation. Repair pathways activated faster. Apoptosis, the polite cellular suicide that prevents mutation cascades, was delayed. In short, human biology, when supplemented with tardigrade machinery, became harder to kill. That was the first tremor. Because radiation tolerance is not just useful for space travel. It has implications for cancer therapy, nuclear energy, military resilience, deep space colonization. It whispers of astronauts who could withstand cosmic radiation without heavy shielding, of tissues that endure chemotherapy with fewer side effects. The grant proposals practically wrote themselves. But biology is rarely a single variable equation. In follow-up studies, something odd appeared. Cells expressing Dsup did not merely show reduced DNA damage. Some lines displayed altered chromatin behavior, subtle changes in how tightly DNA was packaged. Gene expression profiles shifted slightly, particularly in stress response pathways. Nothing dramatic, nothing monstrous, just adaptive. Now combine that with the tardigrade's native cryptobiotic proteins, the intrinsically disordered proteins that vitrify cellular interiors. These proteins don't have fixed structures. They change conformation depending on conditions, forming protective matrices when dehydrated. Flexible, responsive, context-dependent. From a systems biology perspective, this is elegant engineering. The tardigrade genome encodes resilience not as a single shield, but as layered defenses, structural, molecular, metabolic, and layered systems are scalable. This is where the enhancement trials escalated. Researchers began experimenting with multi-gene insertions, plus stress-inducible promoters. Dsup combined with synthetic repair pathway amplifiers. The idea was controlled resilience, activate protection only under extreme stress. But stress is a slippery trigger. In one unpublished dataset referenced in conference slides but never formally journaled, modified mammalian cell cultures exposed to repeated radiation pulses began showing decreasing damage accumulation over time. The first exposure caused expected breaks, the second, fewer. By the fifth cycle, DNA integrity metrics approached baseline even under sustained radiation intensity. Adaptation within clonal cell lines, that should not happen so quickly. Evolution requires variation and selection across generations. Yet these cells, genetically identical, appeared to refine their response in real time. Epigenetic modifications were suspected. Chromatin remodeling, enhanced repair fidelity. Science says plasticity, stress conditioning, known phenomena. And yes, that is the rational explanation. But here's the problem. The more resilient a cell becomes to DNA damage, the closer it edges toward bypassing apoptosis safeguards. And apoptosis exists for a reason. It prevents mutated cells from persisting. Resilience without regulation is a dangerous trait. At this point, the second major climax begins to take shape, not in a dramatic explosion, but in a quiet, data-driven unease. Because the question shifts again. If we borrow the survival toolkit of an organism that laughs at vacuum and radiation, what happens when that toolkit operates inside a complex multicellular system that was never designed for that level of durability? What happens if damage no longer forces shutdown? What if the biological off switch grows reluctant? Officially, no catastrophic outcomes were reported. No runaway cell lines. No cinematic disasters. But several research groups quietly halted multi-gene enhancement trials within the same 2-year window. Funding redirected, focus narrowed, publications softened. The tardigrade returned to its reputation as a charming extremophile. Yet, somewhere in the data logs lies a consistent pattern. When exposed to escalating stress, systems augmented with tardigrade proteins do not simply endure. They stabilize. And systems that stabilize under increasing chaos tend to outlast those that do not. Which brings us to an uncomfortable realization. The tardigrade is not dangerous because it can survive space. It is dangerous because we are beginning to understand how, and we are not particularly famous for leaving such knowledge alone. However, Part 4, the organism that waits here, is where the story stops being about ambition and starts becoming about patience. Because the most unsettling trait of the tardigrade is not its radiation tolerance, nor its glass-like vitrification trick, nor even its DNA shielding proteins. It is its willingness to wait. In laboratory conditions, tardigrades have been revived after years, sometimes decades, of desiccation. In cryptobiosis, their metabolism slows so dramatically that biological time nearly freezes. No growth, no aging, no measurable activity, just suspended potential. From a physics standpoint, this is an extraordinary exploitation of thermodynamic loopholes. Aging at its core is accumulated molecular damage, oxidative stress, replication errors, protein misfolding, all processes that require chemical activity. No activity, no accumulation. In cryptobiosis, entropy is not defeated, but it is deferred. Now consider this in ecological terms. Most organisms survive by competing continuously, reproducing faster, consuming more efficiently, adapting incrementally. The tardigrade opts out. When conditions deteriorate beyond its liking, desiccation, extreme cold, radiation, it simply withdraws. It does not fight the storm, it outlasts it. And that strategy scales disturbingly well in unstable environments. Climate volatility, it waits. Mass extinction, it waits. Radiation spikes, it waits, then it returns. Paleobiological evidence suggests tardigrades have existed for over 500 million years, surviving multiple mass extinction events. The Permian-Triassic extinction eliminated roughly 90% of marine species. The Cretaceous-Paleogene event ended the reign of dinosaurs. The tardigrade persisted. Now, persistence alone is not menace. Earthworms persist. Moss persists. But persistence combined with genomic flexibility is another matter. Recent genomic analyses reveal extensive horizontal gene transfer in some tardigrade species, segments of DNA likely acquired from bacteria, fungi, and plants over evolutionary time. While the extent of this phenomenon has been debated and partially revised, the implication remains provocative. Tardigrades are not static vaults of resilience. They are opportunistic collectors. They borrow, they integrate, they adapt. Picture an organism capable of entering suspended animation during catastrophe, re-emerging into altered ecosystems, and incorporating useful genetic material across evolutionary timescales. Not rapidly, not dramatically, but steadily. This is not invasion biology. This is strategic endurance. And here lies the quiet second major climax. Because while humanity obsesses over immediate existential threats, asteroids, pandemics, artificial intelligence, the tardigrade plays an entirely different game. It is not racing against extinction, it is waiting beyond it. Imagine a future Earth destabilized by climate extremes, increased radiation exposure in certain regions, expanding deserts, fluctuating temperatures. Most complex organisms struggle to adapt to rapid change. The tardigrade does not need to adapt in real time. It can simply pause until the turbulence settles. And if, during that turbulence, human biotechnology continues exploring resilience enhancement, borrowing more from the tardigrade's molecular arsenal, the line between species-specific survival strategy and cross-species integration begins to blur. Again, no cinematic monsters, no apocalyptic swarms, just a microscopic organism that has mastered the art of strategic invisibility. It does not conquer ecosystems loudly. It survives quietly. And in evolutionary terms, quiet survival is often the most powerful strategy of all, because dominance is temporary. But endurance rewrites the future, and we have only begun to decode how it works. Part 5. The data we tried to bury. By now, you may be expecting a dramatic reveal, a classified lab leak, a rogue organism multiplying in orbit, a cinematic disaster complete with ominous lighting and irresponsible scientists running down hallways. I regret to inform you, reality is far less theatrical, and therefore far more disturbing. The nightmare that was buried for 50 years was not a monster. It was a realization. In the early archival experiments, the ones quietly footnoted and never enthusiastically revisited, researchers were not testing whether tardigrades could survive space. That part was already astonishing but manageable. The true question, hinted at in marginal notes and half-finished grant proposals, was whether extreme survival mechanisms could fundamentally shift biological constraints. The assumption in biology has always been that life operates within narrow thermodynamic and biochemical boundaries. Temperature ranges, radiation limits, hydration requirements. These constraints define ecosystems, evolutionary pathways, extinction events. The tardigrade does not eliminate those boundaries. It sidesteps them. By entering cryptobiosis, it effectively removes itself from selective pressure during peak catastrophe. By deploying DNA-protective proteins like Dsup, it reduces mutation rates under high radiation. By vitrifying cellular structures, it prevents structural collapse under desiccation and freezing. Each mechanism alone is extraordinary. Together, they form a blueprint for biological persistence that operates on a different timescale than most life. And here is the buried conclusion that made certain researchers uncomfortable. If an organism can repeatedly endure global-scale stressors with minimal genetic damage and negligible aging during dormancy, it accumulates evolutionary stability while competitors collapse. In chaotic epochs, that is not just survival. That is leverage. Over decades, follow-up funding drifted toward applied biotechnology, radiation-resistant crops, stress-tolerant cell lines, speculative astronaut enhancement. The darker implication was rarely articulated aloud. Resilience technologies alter evolutionary dynamics. Imagine a biosphere increasingly engineered with tardigrade-derived molecular shields, reduced mutation under stress, enhanced repair, suspended metabolic states. Sounds beneficial, doesn't it? Fewer extinctions, greater durability, space colonization made feasible. But evolution depends on failure as much as success. Mutation fuels adaptation. Mortality shapes ecosystems. Limits create diversity. Remove too many limits, and selection pressures shift unpredictably. In one speculative modeling study, largely theoretical, never sensationalized, ecologists simulated a future ecosystem populated by organisms with enhanced radiation tolerance and stress-induced metabolic suspension. The model did not predict apocalypse, it predicted dominance. Not explosive growth, not ecological collapse, dominance through attrition. Species capable of pausing through catastrophe outlast those that cannot. Over centuries, biodiversity narrows toward lineages optimized for endurance rather than speed or specialization. Slow, patient, nearly invisible replacement. Science officially dismisses any notion of the tardigrade as a real and present danger. And strictly speaking, that is correct. It is not stalking humanity. It is not plotting microbial conquest. It is simply exceptionally well designed for a universe that is, by default, hostile. The real danger is not the creature. It is our fascination with its toolkit. Because once we understand how to buffer DNA against radiation, suspend metabolism without damage, and reduce entropy's grip on living systems, we will not resist applying it. And when we do, we may discover that survival at all costs subtly rewrites what it means to be alive. So no, there was no alien contagion, no cosmic horror sealed in a vault, just data. Data showing that life, under the right molecular architecture, can step outside many of the rules we assumed were absolute. The tardigrade is not coming for us. It is waiting. And if history is any guide, it will still be here long after our species has finished arguing about whether it was ever a threat at all.